All right, today we're jumping into something really cool, using artificial intelligence to figure out how cancer patients will respond to treatment. We're breaking down some fascinating work by K. Srinika, G. Aramvalartha Nayaki, and Himalatha Karnan over at the SASTRA School of Chemical and Biotechnology. And believe me, they are tackling one of the biggest challenges in oncology today. Okay, so here's our game plan. First, we're going to unpack the specific problem with lymphoma. Then we'll meet the AI models that are stepping up to the plate. We'll watch them go head-to-head in a performance showdown, see who comes out on top, and finally, talk about what this all means for the future of fighting cancer. Okay, first things first. To really get why this research is such a big deal, we need to understand the enemy, the specific cancer that we are up against. So, the cancer we're zeroing in on is called diffuse large B-cell lymphoma, or DLBCL for short. And you've got to understand, this isn't just any cancer. It's the most common and most aggressive type of non-Hodgkin's lymphoma out there. It's incredibly diverse, which means a one-size-fits-all treatment just doesn't cut it. And here is the absolute core of the problem. For up to 40% of patients, the standard treatment, a chemo regimen called R-CHOP, it just fails. Imagine that. They either resist it from the get-go or they relapse. That's a huge number of people left without a great option, and that is why we need a smarter approach, an AI that can predict who will respond to which drug before a single dose is given. So how do you solve a problem this complex? Well, you bring in the heavy hitters, artificial intelligence. The researchers chose 3 different machine learning models to see which one could make sense of all this complicated genomic data. So let's meet our contenders. Okay, so we've got 3 different models stepping into the ring. First, there's logistic regression. Think of it as the simple, by-the-book statistician. Then you have random forest, which is more like taking a poll of hundreds of experts and going with the consensus. And finally, the support vector machine, or SVM. This one is the master strategist, designed to find clever, even weirdly shaped boundaries to separate data that seems all jumbled up. All right, it's showdown time. The researchers took this massive public library of information, the Genomics of Drug Sensitivity and Cancer dataset, and they basically challenged each AI model to predict how cancer cells would react to different drugs. Let's see how they did. First up, logistic regression. Now this chart here is called a confusion matrix. It's just a report card showing what the model got right and what it, got confused about. And yikes, it was not a good report card. Its simple, straight-line approach just couldn't handle the messy, complex reality of biology. I mean, look at that high sensitivity category. It got every single one wrong. Oof, clearly not the right tool for this job. Okay, next up, random forest. And whoa, what a difference. This is a massive improvement. It got most of the cases right. Its wisdom of the crowd strategy was definitely a better fit. But see how it's almost too perfect? That set off some alarm bells for something called overfitting. It's like a student who memorizes the answers for a test but doesn't actually learn the material. They can ace that test but give them a new problem and they're totally lost. It means this model might look good here but fail in the real world. And this table right here, this is where the plot thickens. Random forest accuracy is over 99%. It looks amazing, right? But now look at its AUC score. AUC is a key metric that shows how well a model can tell the difference between groups. A score of 0.5 is basically a coin flip, and that's pretty much what Random Forest got. Confirming it, just memorize the data. But now, look over at the SVM column. High accuracy, high precision, and a really strong AUC score. It's a rock-solid performer across the board. So yeah, I think it's pretty obvious who won this fight. But the real question is, why? What secret weapon did the support vector machine have that the others didn't? Just let that number sink in for a second. 99.88% accuracy. That is insane. It's practically perfect at predicting how these cancer cells will behave. And it did this thanks to a special trick up its sleeve called an RBF kernel. So, what on earth is that? Well, this image shows you exactly why it worked so well. The other models tried to draw simple, straight lines to separate the data. But look at the SVM in the bottom left. That RBF kernel allowed it to draw these complex, curvy, flexible boundaries. It didn't force a simple answer onto a complex problem. Instead, it found the true, intricate patterns hidden in the genetic data. It was basically speaking the language of biology. Okay, so SVM is the champion. That's awesome. But what does this victory really mean for patients and for the future of treating cancer? Let's zoom out and look at the big picture. So, a few big takeaways here. First, the right tool for the job matters a lot. The SVM model with that specific kernel was the key. Interestingly, it also found that one single feature, a measure of a drug's raw power, was the most important predictor. But, and this is a really important but, we have to remember this was all done in a lab with cell lines, not real people. Plus, these models can be black boxes, and getting doctors to trust a prediction they can't understand, that's a whole other challenge. But at the end of the day, this research is a huge step forward. It powerfully backs up the whole idea of precision medicine. Like the authors say, the goal is to use these tools to create targeted treatments and ultimately improve patient outcomes. It's all about finding the right drug for the right person at the right time. And that really leaves us with the big multi-million-dollar question, doesn't it? We've seen what this AI can do in the lab. The predictive power is incredible. So what's it going to take to bridge the gap from the lab to the clinic and bring this kind of power to every single patient? That is the challenge that researchers are tackling right now.